Hi, it's time to talk about writing a discussion section. So, the whole point of a discussion section is to have you take the results that you got and fit them into the big picture of science. Remember, science doesn't happen in a vacuum, and so we're always trying to expand our understanding of the world by a little bit every single time. It's rare anyone makes a huge leap in one direction or another. And so, what we want to do here is say, not just here's this thing that we learned, but where is this thing that we learned relative to all the other things that we already know? And essentially, what we want to find out is what exactly do your results mean for the larger picture of science? Now, that statement can be misconstrued. When you talk about what your results mean, we don't want you to talk about your opinion necessarily of what the results mean. So if you make a statement about what something means, you need to back that up with evidence. Every single conclusion you draw has got to be rooted in the evidence your results present. So when you do that, you want to avoid value judgments. And it's rare that you think about science as having value judgments of good or bad, and that's for a reason. It's really hard to say, this is really good data. We don't want to hear that. What do we want to hear? We want to hear some sort of numerical data that proves to us how good or how bad your data were. So before you start looking at the bigger picture of science, start off by looking at just your data and try and coalesce your data into something that's a little more compact. So try and see how things fit together. If you measured multiple different things, how do they work together? So if you want to do ANOVA or whatever, this is a good place to do it. You want to talk about the limitations of your method. So this is the part that we normally talk about in lab as error analysis. But you have to be pretty careful about this because you don't just need to come up with what could have gone wrong. You also want to talk about how likely it was that that thing went wrong. And in fact, a lot of this error analysis we do before we ever get to the point of actually publishing a paper, because usually we'll do that error analysis, and if there's anything we can fix, we'll redo the experiment fixing that thing, right? So there are some things that we can't fix. We might be measuring near the limit of quantitation for our analytical instrument. That is something we can't fix, and there may not be an instrument with a better detection limit, so we might be stuck, right? That might be the best we can do. We might have factors that preclude us from getting as much data as we want. For example, you can do a transect measuring the chemistry of the ocean, from Honolulu to San Diego, which is something that I did, but you are not measuring every single inch of ocean between here and there. You're not even measuring every kilometer of the ocean between here and there. What are you measuring? Maybe you'll do one measurement every 100 kilometers or 200 kilometers or something like that, right? So there are limitations on how you are measuring. One of the ways we can evaluate those limitations is by looking at reproducibility. So looking at things, for example, like a standard deviation. So a lot of times the standard deviations are just reported as part of the data in the results section. But if you have large variations, what could be accounting for that? Okay, and here is where you start drawing in that larger context. Okay, if you're measuring something about bacteria and everything else about bacteria is that, as conditions change, their chemical output changes, and you do tend to get large variations in what happens, then maybe, you're seeing a large range, not because your measurement ability is bad, but because bacteria do a lot of weird different things. So the way that you do this, you're comparing your results to those found in previous studies. Now, if you think back to what I said in the introduction, odds are very good that there is not something that is specifically about the thing that you in particular are studying. What you want to do then is you want to compare it to other studies that have been done. So, for example, let's say that you're looking at toxic red tide algae, and you discover that they produce really high amounts of the free radical superoxide, and that other organisms that people have measured superoxide for are far, far less than your measurements. Well, what could you say? Well, yeah, these make a lot more. Maybe that's why they're so toxic, right? So you're putting everything into a context. Does your data match up to what we know about other things? How does it relate? If it doesn't, is there something about what you're studying that makes it stand out from everything that's been studying before? Does that difference explain the difference that you see in your results? If you started out with a hypothesis, does the data support it? I said before that we don't always explicitly state our hypothesis in the introduction section. Your data may or may not support whatever the prevailing wisdom is in the scientific community. It was thought that most hydrogen peroxide in natural waters was produced through photochemical means. But what we found was that it also occurs in the dark. Here, your data do not support the hypothesis. What explanation, then, do they support? This is where you need to make a statement. And remember, this is not an opinion. You make a statement, draw a conclusion that is backed up with evidence. Now, finally, you're going to talk about questions that are still unanswered. You usually start a study saying, these are the questions I want to answer in this study. And if you have 3 questions, you're lucky if you answer 1. And usually that study will also generate about 50 more questions. So the discussion is a good place to point out things that are still unanswered. And the reason we do this is because, science keeps going on and on. And so we often need to think about, okay, what else can I do? What is your next study going to be about? Probably these questions that you still haven't answered that are probably driving you crazy. So now let's go to those papers that we always look at, and let's see what they do in the discussion. Okay, so we're back to the Rowe 2012 paper. And once again, you'll see they've broken this discussion down into different sections that each discuss an aspect of the study that they did. The first thing they do is they talk about their particular study in the laboratory. Why was that good? There's a less complex environment to study the iron acquisition strategies. Now, if you look at this part right here, our data indicate that trichodesmium under our iron-limited culture conditions can most readily access inorganic iron chloride, or iron in association with weak organic ligands, iron citrate. Acquisition of iron from strongly bound organic complexes was at least an order of magnitude lower than that of the iron citrate and iron chloride. Now, if you look back at the results section, you will see that table where they showed you how much each thing took up. Here, they're telling you what they think those numbers mean. Now, in the next paragraph, here's what they say. Previous work on cellular metal cycling by nitrogen-fixing cyanobacterium has shown in iron-replete conditions that trichodesmium and metal concentrations do not vary over the diel cycle. So what they're doing here is they are going back to previous studies. You'll notice that unlike in the previous paragraph, suddenly we have a whole bunch of citations. Here we have Tuitt et al., 2004. Castorita et al., 2007. Saito et al., 2011. Now we have a paragraph about bioavailability of iron species. Remember, we're talking about iron bound to what? Iron chloride, iron citrate, all those other things. Now they're talking about what other people have noticed about the bioavailability of various iron species. Chappell and Webb, 2010. Hopkinson and Morrell, 2009. And so they have all these things that talk about what is known about iron acquisition, and they're putting their results in the context of what was known about iron acquisition in various organisms. Not all of these were trichodesmium experiments. You'll see they do the same thing for the acquisition of iron by representative associated bacteria, and I'm not going to go into it. It's a fairly similar thing. Here is what I want to look at. The implication for trichodesmium colonies. Okay? So, trichodesmium colonies are a complex microenvironment in which diverse organisms thrive. And so now what they're going to do, they have experiments on trichodesmium iron uptake and experiments on the bacteria associated with those colonies, because bacteria love to hang out on trichodesmium colonies. Now, what they're going to do is say, how do these things work together in the wild? Possible mutualistic relationships between bacteria and trichodesmium facilitating iron uptake are of interest. Note the citation, Kuska. The ability of trichodesmium colonies to collect dust particles, 2 references, and the ability of bacteria to produce siderophores, which have a pronounced effect on the dissolution kinetics and the solubility of iron-bearing minerals, another citation, may provide a source of iron that both organisms can utilize. So they've gone through, here are things that we care about out in the ocean. The uptake results presented in this study agree with this Achilles study. And now, however, we found that, so here's a study. What we did reinforces what this study found. But what we did also found this completely different thing that the other study didn't find. Now, here's something that's interesting that you may not have thought about in regards to a discussion section, but it happens in a lot of discussion sections. To better understand how these organisms may be competing with each other in a colony environment, we modeled trichodesmium colony uptake using the iron uptake measurements obtained for individual organisms in this study. What does this mean? They measured rates for single organisms. They also know how those organisms live together in what approximate proportions, and now they're going to look at what is a whole community going to do. They are not going to measure a whole community. They're going to take their measurements for individual community members, and they're going to create a model for how a whole community would work. And so they clearly are going to come up with a number, and you can see that they've illustrated these numbers with a figure. So it's perfectly good to have a figure in the discussion section. That's actually very common. Okay, so what do they have? They have the 2 types of iron that are easily taken up, iron citrate and iron chloride. And what they have here is they have this white part that shows how much the trichodesmium is taking up, and the black part that shows how much the associated bacteria are taking up. So what they have here, A, B, C, D, are different sorts of model colonies, right? Because every model you make has assumptions, and what if your assumptions are wrong? Well, they just tried a whole bunch of assumptions, and now they're showing us what those assumptions would look like. Here's a table that shows similar things. Now, here's how they finish it. The findings of the study suggest that, although they may share the same microenvironment, trichodesmium and its colony-associated microbial community may differ substantially in terms of iron acquisition strategy. So that is their final takeaway. Okay, now let's look at the Lynch paper. Discussion and interpretations. Once again, we have multiple sections in here. We have sulfates, phyllosilicates, and there's more. Sulfates, they talk about gypsum. Now, if you go back and you look at the results section, the results are broken down by the methods that were used to analyze the minerals. They use QuemScan. They use XRD. They use VNIR. A whole bunch of stuff. What they're doing now is taking all the results from those different methods and amalgamating them to say, hey, there's sulfates and it's mostly gypsum. Hey, there are phyllosilicates, but how are they distributed throughout the valley? And so we see no spectral evidence of these minerals that are abundant in the middle of the valley. Therefore, what happens? They might be buried under a whole bunch of stuff. How about carbonates or microbial mat, the thing that we are very interested in? Other salts. So you can see that basically everything's being gone through analyte by analyte. And now we get to the important thing, the summary discussion of spectral interpretations. And you can see here is where we start pulling in those other references. As stated previously, minerals rarely form as pure end members. Okay, so here's Clark 1999, Bishop 2013, Clotis 1986, Horgan 2014, Singer 1981, Stack and Milliket 2015. So whole bunch of references. More laboratory studies on highly complex mineral assemblages are warranted. What does this tell us? We need to know more about these mixes of minerals. That's basically the takeaway. The biggest effect on mineral diversity is that, and here's the explanation of what we found. I keep switching between we and they. This was something I helped on, so. Implications for Mars, because this is a Mars analog paper. Why is this important? Here's a summary of conclusions. Analysis of the VNIR spectra alone is insufficient. What they're saying is, you can't just throw satellite overhead, look at the planet, and say, yeah, now we know all about it. You actually have to go down and dig down, right? You got to send down the rovers. You can't just throw the orbiter in place. More understanding of mineral mixing is required. Location matters. And more understanding of chloride hydrates and chlorine oxy anion cells is required. So when they say more understanding is required, what does that mean? Hey, we got to do a bunch of experiments. Some papers will actually have a dedicated section for future work that should be done. Here it's called out by saying, this is what is required. So essentially what you're doing in a discussion is you're telling your reader how your results fit into the big picture of what science already knows. Also, to an extent, what science still doesn't know, right? The whole future work thing. That's what you're shooting for in a discussion. A reader should understand what the scientific community learns from your results, how it fits in with what others have done, and what should be done in the future. I hope you found this helpful. I'll see you again soon.